Hello, I'm Dr. Aizaz from MedicoVisual.com, and in today's visual lecture, we will talk about the development of hair. Now, here we have the stratum basale. I'm just showing the stratum basale here, and below that we have a piece of developing dermis. So, let me show you here. So, we have a piece of developing dermis. Actually, outside the stratum basale, there is another flattened layer of cell, and this is called periderm, which I haven't shown at this in this 3D model. Now, at this two cell layer stage, which is periderm and stratum basale, what actually happens that the cells of stratum basale, they protrude downward at a particular point. They protrude downward, or they develop a downward extension into the developing dermis, or we can say that that extension invades the developing dermis, and this structure is called hair peg. Let me show you that. So here you can see this is the hair peg. Now the lower end of this hair peg, it dilates to form the hair bulb. So this part is called, this is the hair bulb. Lower dilated end is called, this is the hair, this is bulb like structure, it is called hair bulb. Now this hair peg is actually the precursor of the hair follicle, which will form the hair, the strand of hair, the shaft of the hair. Now to understand the structure, what I will do is that I will take a section of it. What I mean by this is that I will cut it from here like this. I will cut it from here, and then we will see what is going on inside. So here I have taken the section. Now you can observe this downward extension of stratum basale into the developing dermis. Now initially the dermis was invaded by this stratum basale, the hair peg or hair follicle. Now it's time for the dermis to take the revenge. So what dermis will do is that dermis will invade the lower part of hair follicle. It will invade the lower end of the hair bulb from the downward side like this. So you can see that here there is this inward extension of dermis into the lower end of hair bulb or hair follicle, at the base of hair follicle. This extension of dermis into the hair bulb, it is termed as dermal, let me write it here, dermal hair papilla. So this is dermal hair papilla. Within this dermal hair papilla, blood vessels develop, and these blood vessels, of course, they will provide the nourishment to the developing hair. Now later in this lecture, we will see that somewhat from this region, the sebaceous gland will develop. Somewhat from this region, the sebaceous gland will develop, and somewhat here a smooth muscle will attach, and this smooth muscle is called Arrector Pili muscle. Now this region, this is the region of origin of duct of sebaceous gland, and this region is the site of attachment of Arrector Pili. Between these two regions, somewhere in midway, somewhat here, the cells, the stratum basale cells here. Now actually these are not the cells of stratum basale, they should now be called the cells of hair follicle. So here the cells of hair follicle, they will proliferate and they will form an aggregate of the cells, a bulge of undifferentiated cells here, that will be formed like this. And this bulge is called, very simple and logical name, it is called hair bulge. It consists of undifferentiated hair cells, and all the layers of our hair shaft and some other layers that we will just talk about, they develop from this hair bulge. Well, actually, hair will arise here. Hair will arise here. Yeah. So hair will arise from here, but hair bulges here. So what actually happens that these cells, they will migrate from here and they will come to lie in this region. So they will migrate from here. They will grow, proliferate, and migrate from here like this, and come to lie in this region. Here, now this aggregate of undifferentiated cell in this region, this is called hair matrix. This is not the matrix that Andrew Tate talks about. This is the hair matrix, right? No, this hair matrix is the precursor of all the layers of hair and some layers of the hair follicle as well. So, but before that, we also need the blackening agent, the melanin pigment. So, neural crest cells come and they convert to melanocytes in this region, and they form the melanosomes, which will deliver the melanin pigment to these hair matrix cells. And these hair matrix cells, when they will proliferate and they will form the hair at other layers, those layers will of course contain the melanin pigment. So ultimately those layers will be formed like this. Now, actually, this is almost all about the development of hair. But beyond that, I will briefly talk about the histology of hair. Of course, I will not go into detail, microstructural details, but some basic histology that I will talk about. And after that, we will also discuss the development briefly about the development of sebaceous gland as well as the erector pili muscle. Now, if I zoom in, what you can see here is that this is the actual hair strand here. So hair strand consists of three layers. This red one here, of course, it is not red. Here is black, but this is just for demonstration purposes. The red one is the medulla. Outside orange one is the cortex, and outermost is this flattened cell layer. This is called cuticle of hair. This is called cuticle of hair. I have made it translucent so you can observe some structure details, but I will not go into the exact microstructure details in this lecture. We will discuss those in histology. Before going further, I want you to focus here. Here you can see that you can see the nucleus up to this point. You can see the cells are filled with nuclei up to this point, but beyond that they are not filled with nucleus. There might be some fragments of nucleus, but there is no actual complete nucleus in those cells. So cells beyond this point, these are actually not cells, these are dead cells. Dead cells, these are basically dead bags of, these are cell membrane bound bags of keratin protein. So keratin began to form from very beginning. So keratinization start, but above this, beyond this point, the cells are also dead. Beyond this point, there is cornification. In the last lecture, I did a mistake. I mixed these things up. Actually, it was a tongue slip. So there is a difference between cornification and keratinization. So cornification is not similar to the keratinization. Keratinization, in simple terms, it simply means filling up of cell with keratin filament, right? But cornification, on the other hand, it is a special type of programmed cell death. Normally, when the cell die, if I draw a cell here, normally when a cell die, as the nucleus will die, the nucleus will fragment and die, eaten up by macrophages, and of course organelles will also die. The cell membrane will also die. The cell membrane will also rupture, and ultimately the cell will be disrupted. But here the cell membrane is intact. Cell membrane does not die. Only the nucleus die, organelles die, nucleus and organelles die, and it gets filled with keratin filament. So it is a membrane bound bag of keratin filament, and the keratin filament of hair is different from the keratin filament of skin. The skin's keratin filament is soft keratin. This one is the hair's keratin is hard keratin. I will not go into microstructural and biochemical details. That is the topic beyond scope of this lecture, but you get some basic idea. Now beyond this point, there are no living cell, but the base of hair is living. So that is why when you go to barber and get a new haircut, he or she cuts your hair, you don't feel any pain. But if you pluck your hair, if you pull your hair out from this root here, this is the living part, it is also surrounded by nerves. So then you feel some pain. That is important to understand because this part is living, but beyond that, it is totally dead cells. So this point above which the cornification start, below this there is only keratinization. Above it there is keratinization as well as cornification. Keratinization is that cell is filled with keratin, but along with the filling of keratin there is specialized apoptosis, specialized programmed cell death, in which the nuclei as well as organelles that also die. So that specialized programmed cell death is cornification. The cell membrane remains intact, but nuclei and organelles die. That is cornification. And this somewhat here, this region is called Adamson's fringe. I think most of you don't need to remember this, Adamson's fringe. But if you want to remember, you can remember that this is called Adamson's fringe, right? So yeah, so now briefly let me label the layers of the hair strand as well as the layers of the hair follicle. So about the hair strand, basically I have told you already that the deeper one is medulla of the hair, and it is not present in all type of hair. It is present in the thick hair, and it is actually filled with air as well. So this is not present in thin hairs. And outside it is the cortex of hair, and outermost a thin layer of you can say diagonally arranged flattened cell that is called cuticle layer. So all these three layers are the layers of hair shaft or hair strand, hair shaft. Now if I come more superficially, this purplish one is a thick layer, and it consists further of multiple sublayers. I will not go into details of those sublayers. That is the topic of histology. We have Hensley layer, Huxley layers, and cuticle, different layers, 3 layers basically. Let's forget about them right now. So this is called internal root sheath. Internal root sheath. Then if I come further outward, we have this layer which is continuous with what? Which is continuous with stratum basale. This is the one which actually formed this hair follicle. The other layers, they arise from this matrix, the cell matrix. But this layer, it is direct extension of stratum basale. It does not arise from hair matrix. It is a direct extension of stratum basale. This layer is external root sheath. It's very logical. We have internal root sheath, then of course we must have external root sheath as well. And both these layers, these are epithelial in nature. I haven't shown the exact epithelial nature of the internal root sheath, but for now, just trust me that it is also epithelial. So both of them as a whole, if we talk about both of them combined, then if we consider them as a single entity, then this layer as a whole is called epithelial. Very logical name, epithelial root sheath. Epithelial root sheath. So if we have epithelial root sheath, then it means that we must have a fibrous root sheath as well. Well, actually, as it turns out, we have a fibrous root sheath as well, and the fibrous root sheath arises from the developing dermis cells of the developing dermis. It is basically a condensation of connective tissue. So this greenish one here, this condensation of connective tissue or developing dermis, it is called, this one was epithelial, so it is fibrous. It is fibrous root sheath, or we can simply call it fibrous hair sheath, or we also call it dermal hair sheath, or we also call it connective tissue hair sheath. So it has many different names, but all these are very logical and you can understand what I mean by this. And by the way, there is some confusion that some students have. You know, in textbooks, somewhere in some of the textbook, you will see that the dermal hair sheath, they write it like this, that dermal hair sheath, it arises from mesenchyme. Somewhere they write it arises from mesoderm. Then somewhere they write it arises from dermis. So you people get confused. I mean, whether it arises from dermis or if it arises from mesenchyme or mesoderm. Well, actually, all these three terms mean the same here in this context. The dermis, it arises from the mesenchymal cells that arise from the mesoderm. So we have this dermis, and this dermis, it itself came from mesenchymal cells, and these mesenchymal cells, they themselves came from mesoderm. So if you say dermal root sheath or hair sheath, it arises from mesoderm, that is also correct. If you say it arises from mesenchymal cell, that is also correct. And if you say it arises from dermis, then that is also correct. Basically, not dermis, I should say developing dermis. So this is a basic idea. So here you can observe that hair sheath consists of these three layers, but hair sheath is connected and it is glued together, it is bridged together, it is tightly adherent together with the walls of hair follicle. These are the walls of hair follicle which are formed by external root sheath. So these are adherent, these are glued together with the walls of hair follicle with the help of this internal root sheath. It's a tongue twister for me. So this internal root sheath, which itself consists of multiple layers, 3 layers, it connects the hair shaft with the walls of hair follicle. So it was important to understand. And by the way, let me tell you that this internal root sheath, it does not reach all the way to the top. Here actually there will be the development of sebaceous gland, and at near the duct of sebaceous gland, here it terminates and continues with the wall of sebaceous gland like this. So it is continuous with the walls of sebaceous gland. It does not reach, this layer does not reach up to the top. Please understand this thing. And also please note that the external root sheath, these cells may proliferate and they may form more than one layer. Here I have shown a single cuboidal cell layer, but more than 1, 2 or 3 layers may be there. Please understand this thing. And of course here complete above it stratum spinosum will develop, and stratum granulosum, other cells will develop. A complete skin epidermis of skin will develop here, which I haven't shown in this 3D model because our focus and our topic of discussion was the hair follicles, right? So now let's talk about the development of sebaceous gland. So as I've told you, at the upper extent of the internal root sheath, somewhat here, there is another evagination of the cells of the cells of the walls of this pilosebaceous unit. Pilosebaceous mean pilo mean hair and sebaceous means sebaceous gland. So both of these structures they form a unit. So the stratum basale of this pilosebaceous unit, it evaginates again into the dermis. Another invasion of the dermis occur, and that's how the gland develops like this. I don't know why the stratum basale is doing so many invasions of the developing dermis. It is not even rich in oil. So here you can see this gland develops. Now if you focus on the structure of this gland, if I hide the dermis, we can focus on the structure of this gland. Now this gland, it consists of, let's draw a simple diagram to understand it. So this gland has a single duct. This is the duct of this gland. So this is a duct of this gland, and then its secretory component, it is divided into different units, grape-like or alveoli-like units, right? So these grapes-like globular units, the secretory part, these secretory parts here, these secretory parts, these are not tube-like, these are globular, these are acinar-like, these are called acinus. So this type of gland is called branched acinus. So we have branched acinus gland, branched acinar or acinus gland. And this is the structure of sebaceous gland. It's very easy to understand. This gland secretes an oily secretion, but we will not discuss its physiology, that how exactly it secretes the oily secretion. And then what actually happens that some of the mesenchymal cells of this region, or mesodermal cells, or cells of developing dermis, some of these cells, what they do is that they will collect here like this, and they will form smooth muscle. This smooth muscle is called Arrector pili muscle. This is the Arrector pili muscle. So here we have a hair in its beloved hair follicle, and here is this Arrector pili muscle. Please understand that initially as the hair peg and hair follicle develops, it is at an angle, it is diagonal, it is not straight or perpendicular, it is not perpendicular downward into dermis, it develops at an angle diagonally. So that is why here is the attachment of Arrector pili, and as this muscle contract, the hair follicle sort of straighten up, causing the hair to stand erect like this. So initially the hair was bent. Initially the normal position of hairs is that it is bent, but with the contraction of Arrector pili muscle, the hair becomes more straight or more erect, straightened up like this. It has many function in animals. It has some role in thermoregulation, that we have, let's say, hairs like this standing up. So they may trap some air here, and this air may act as an insulator, right? So it has some role in thermoregulation. It has some role in prey instinct, that it makes the prey, a bit larger looking, a bit bigger looking. So it has some functions in animals, but it is said that in humans it is mostly vestigial. There might be some role in thermoregulation, but it is not very important. And by the way, this process, in simple English we call it development of goosebumps, and the technical or scientific or medical term is the piloerection of the hair. And that is due to the Arrector pili muscle. So that was all about it. The first hair that are formed of a baby, a newborn baby, these are fine and these are not very well pigmented. These are not very mature type of hair. These are not well built hair. So those hair that are called lanugo hair. I hope I am pronouncing it right. Lanugo hair. So those initial thin, flimsy, and not very much mature, less pigmented hair, these are shed at birth, and then later they are replaced by more better and more mature, coarse type of hair later. Right, so that was all about the development of hair follicle, sebaceous gland, and erector pili muscle. Thank you so much for watching this video. Have a great day. Goodbye.